So the purpose here today is to review the basic concepts, some underlying themes of the summer assignments. I want to make sure we're on the same page, and it's something we did in class, but some people wanted to hear it again on their own. So I do this once in a while, and it seems to be helpful. So let's get started. So we're talking about mitosis versus meiosis, and we're talking about evolution and chromosomal line of inheritance and Mendelian genetics and all these words in between. And I'm not here to go over everything, but give you how these things connect. So let's start. Now let's say I say 2n equals 4. We should realize that this valuation or this expression is meaning something called the diploid. The diploid number is the actual number of chromosomes that the organism has. Now I say organism because an organism is made up of chromosomes, which are made up of homologous. Why there's a little H there, so homologous. Now, what's a homologous? Well, they're the same chromosomes. They come in pairs. Now, we know that the human, all right, has a 2n number equals 46. It has actually 46 chromosomes. Now, what it really has is, okay, 23 homologous pairs. Now, what a homologous pair is are two of the identical type of chromosome that carries the same type of information or gene or allele and expression. Okay, they could be different. Okay, so let's talk about that. So let's talk about one allele. Now, allele, of course, is an expression of a gene. Now, what does that mean? Well, it's usually something we can see. For instance, we talked about in class, if you talk about a flower, and I'm not really good at drawing flowers or anything in between, but if I have a flower that's red versus a flower that's white, and I'll just put red or white, we should be aware that these are called alleles, and that's white. So I could have a red or a white flower as a, let's say, an expression of a certain type of gene. And now what's the gene? Now the gene is an area on the chromosome, okay, where the information, the genetic information, is stored to make the flower either white or red. Now let's think about this. If we have two chromosomes, and we always have pairs in a correctly working organism. This is, of course, the nucleus, and I'm just drawing these as the centromeres. Now, this is a homologous pair right here. What's so special about this pair is that we have a maternal and we have a paternal, meaning from the father, meaning one of these chromosomes came from the mother and father. Now, how'd that happen? Well, in fertilization, okay, a gamete, which has half the number, in this case, if 2n equaled 4, n would equal 2. Okay, and we'll get back to this designation in a second. So if I'm talking about a pair, a homologous pair, one came from the mother and one came from the father. Okay, the father's sperm, right? This would be a gamete, right? The gamete is what has half the number. And this would be the egg. And they came together and they made a homologous pair. And what they have on the chromosome is areas, and I'll just make an area in the chromosome where there is a genetic code. And that genetic code is DNA. Deoxyribonucleic acid, DNA. Okay? And what it really is a sugar, a phosphate, and a nitrogenous base. And those nitrogenous bases, okay, are really what the code is. You have, A, and you could have a G, and you could have, let's say, a C, and these are adenine, guanine, cytosine, and these are the nitrogenous bases, and they would pair up appropriately. And we'll talk about this. Uh, and they have a code, and DNA is double stranded, and there's an appropriately pairing of this code. This code right here, this genetic code, which is what a gene is made up of, okay, will make something called a protein. All genetic codes eventually make proteins, and this protein, okay, makes or basically becomes an enzyme, and we'll learn about all these specifics, which makes a chemical reaction occur or catalyzes reactions, and the chemical reactions that occur make the trait. For instance, if I'm dealing with a red flower, there's red pigment, red chemical, okay, that's responsible for the color in the red. So therefore we have to make it. So in order to have a red flower, to show and express that trait, which means we have the allele, the red allele, we have to have a chemical reaction that makes it, which means you have to have the protein that makes that chemical reaction go or catalyzes it. And that, of course, is coded for in the DNA in a certain spot in the chromosome. So that's the chain of command. The chromosome has the code. This actual code will code for the individual units of a protein, these amino acids, and they build them to make the protein, which will fold into a certain three-dimensional structure that will actually make the reactions go to make, in this case, the red pigment. And so that's how it works. Now, we talked about one of these chromosomes coming from the mother, the egg, and one coming from the father, and fertilization makes the organism. This is the diploid. This is where you have both pairs, okay? Now, of course, there's going to be, okay, 22 other pairs of chromosome, but we're just focusing on one. In fact, I'm talking about the pea plants is far less than 23 pairs. I think there's 9 or so, and don't quote me on that. But so it's much simpler than us. But in any case, back to this pair, this homologous pair. The alleles, again, they have the same type of genes, which, now what's a gene again? A gene is a stretch of DNA code that actually codes for a certain protein. So it's in a certain area. Now, when we talk about diploid number and the actual numbers, we always think of pair. You'll never have a diploid number of 5 or an odd number. It's always going to be an even number in a normal organism. Certainly there are some scenarios where they're not. Now, when I talk about a haploid, we're talking about the gametes that make them, and we're going to go back to that. But I want to talk about something you saw when you dealt with all of this, and we talked about the dominant allele, and you talked about the recessive. Now, let's make red big capital R and recessive lowercase. We put this together, what do we get? We get R, little r. And this is called genotype. This is genotype. Now, why they call it genotype is because it tells us essentially the type of genes that we have available. We have a gene on a chromosome. Let's make this one big R. And then we have another gene for the small r. So this is telling us the type of gene. So we have a stretch of DNA that will express the red pigment, and then we have a stretch of DNA that will not. That's the recessive. Okay, and some people learn that, hey, in order to have a recessive, you have to have a genotype that's homozygous. Right, like that. But you're always going to see pairs in genotype because guess what? We have pairs of chromosomes. Now, Mendelian genetics, where we got our first understanding, okay, he did not know, the monk there, Gregory Mendel, did not know about chromosomes. but he started realizing that there was dominant and recessive. He was onto something when he figured out there was a trait that dominated another and they came in pairs. So he was laid the groundwork, okay, for these letters to represent spots on a chromosome. But I need you to always understand chromosomal line of inheritance is happening on his chromosome. Okay, so what does this mean? Well, we know from our reading that I can have a genotype that's big R, big R. This is homozygous dominant. And we can have big R, little r is heterozygous. And we know that these two will give me my red phenotype, right? And this is the only one that will give me white. Now, genotype is the type of genes. Works back to the type of gene on a maternal or paternal chromosome. The phenotype, a lot of words here, is the type of expression or allele you see. We have a white phenotype or a red, and we can see that we can have the same, we can have multiple genotypes for the same phenotype. The question is always going to be why? Why is it good enough just to have one dominant R? Why did Mendel call this dominant? If I had a big R and a little r, it was going to be red. And the answer to that question is, the recessive doesn't like back down to the dominant. The idea here is that the red is a working gene on a chromosome that correctly codes for a protein that catalyzes a series of chemical reactions that makes the red pigment. It's the working gene. The recessive gene is a broken gene. This one's broken for some reason, and probably because it has a mutation, meaning somewhere in its genetic code there's something that's not correct. We'll learn about mutations, but the base pairing here is wrong. A mutation could occur, and maybe instead of AT, it's AG. It's incorrectly paired. And this code that's incorrect will code for a protein that has a different three-dimensional structure that does not work the same. The reason why these enzymes can make reactions occur is because they have certain three-dimensionality. So this code makes amino acids, and we're going to have different fundamental units of this protein being made, which will make it a different shape, which means it won't make the same reaction go. And so when you think about that, if I have a homozygous recessive, little r and capital R, this is still going to give me the red allele. Why? Why am I still going to make the red pigment? Well, this chromosome doesn't code for the pigment, but I still have a chromosome that does. So I still have one chromosome that can correctly code for the trait, meaning makes the red pigment. That's why homozygous recessive is still red. Obviously, two big R's, both big R's here would mean that both code for the correct protein that makes the chemical reactions go, that make the pigment. But here, and this is the key, understanding why, homozygous recessive, I have two R's. And these two little r's are broken, so both of them combined, there's no other big R. So if there's two little r's here, pretend I'm a princess and this is a little r. So if you got a little r here, you don't make the what? You don't make the red allele. You make a protein that doesn't make the reactions go to make the red. In the absence of red, you've got a white flower. And the only time you have a white flower is if you do not have a working red allele. That's why it works. So the only way to get the white phenotype, the genotype, has to be homozygous recessive. And that's a classic dominant recessive Mendelian genetics that he found. And he had these. He didn't understand any of this right here. None of this was known yet, but he opened the door for that. Understand, he was able to do Punnett squares and predict by reacting these like we're going to do in our fast plant lab, because essentially with these very simple traits, okay? It was basically just separating this allele or this allele. So if you have a organism that's going to make a gamete in meiosis, and that's important you understand, all right, you could have a gamete, okay, remember that's going to have half. It's a haploid number. It's going to have, let's just pretend very simple, 2n equals 2. That's just one pair. I'm going to have one chromosome, either the mother or the father. You don't know which one, and the chances of getting them is 50%. It's 1 out of 2. It's flipping a coin. Now, if you think about humans, we have what? 23 pairs, okay? So every one of those chromosomes that we get when we make a what? Sperm or an egg. Now, we're not really an individual, we're a gamete. We have half of them. They are half that number, and when you make a gamete, let's say a sperm cell, the male version of the gamete, you're starting with a complete organism, and you're going to put in there half of the chromosomes, but you don't know if they're going to be half of the what? The mother or half of the father or some combination. And that gives us something called genetic diversity. But before we get ahead, let's talk about this number and what it looks like. Okay, let's make our 2n equal 4 again. Okay, so this tells us that we have 4 pairs. So in my regular cell in the nucleus, I have 4 pairs of chromosomes. Why is it an even number? Because we have mommy and daddy chromosomes, one from each, that made the organism. Now, okay, and they came together to fertilize the egg. This is a zygote. This is an individual with the complete set of 4. Now, what's confusing, and we're going to do mitosis first. What's confusing is how we start, okay? If 2n equals 4, okay, and I asked you to draw something called the prophase. The prophase is the first stage in mitosis or meiosis. And let me just talk about the cell cycle here for a second. Here's the life of a cell. And if you look at the life of a cell, okay, it's broken up into a couple of different sections. And these different sections help us understand how this cell spends most of its time. So if we do, like, say, put some arrows in the show, it's clockwise fashion, okay? And this little section right here would represent mitosis. Now, what's mitosis? Mitosis is this complete duplication. Has nothing to do with sexual reproduction. We want to make an exact copy. Now, why would we want to do that? Well, because some cells in our body have a shelf life, meaning they only last a certain amount of time, and eventually they lose their functionality and their efficiency. A great example is the red blood cells in our body that last about 60 days. They deliver an important element, oxygen, for us to survive, and without them working appropriately, we would not. So we make sure that we always have fresh red blood cells efficiently doing their job. But what's interesting is that when we do mitosis, maybe it's not interesting, that only a small part of the life cycle of the cell before it dies, okay, is undergoing duplication. So what we have is different phases. And I'm just going to break this up. You don't have to know this yet, but we will get to this. This is the G1 phase. This is the S phase. And this is the G2 phase. Bottom line is, the rest of this cycle, if I was to shade it here, green, I don't know if that's going to work, but the rest of this cycle is interphase. Interphase is the rest of the cell cycle. Now, what's an interphase? Well, this entire life of the cycle spends most of its time in interphase, and this G represent growth. And basically the G1 phase, and you have the S and the G2 phase. Now, what you have here in the G1 phase is you have the cell growing and doing its job, and basically it's growing to get bigger, and it's doing whatever job it has. If it's a beta cell in the pancreas, it's making insulin, and so different cells have different jobs, and it's growing and doing that job. Now in the S phase, this is still interphase. What's happening is that the chromosomes, or I should say the DNA, is duplicated. Yes, Okay. Now it's duplicated. That doesn't mean it's mitosis, because mitosis is making a brand new copy of the original cell. All right. So what we have here is that we duplicate the DNA, which means the chromosomes get duplicated. And then G2, the cell continues to prepare for mitosis, but mostly it's doing its job. And basically, it's also checking for errors on this duplicated. So most of the time, DNA is in its basically not really in a chromosomal form. And let me explain. Okay, when you learned about prophase, you learned that DNA was condensed. And what does that mean? Well, it means, and let me just make some room here because I can. What it means is that when we use the DNA, remember, most of the interphase, the growth cycle of this cell, okay, most of the cell's life is in this stage here. And what we have interphase is it's doing its cell job, and it's growing, and it's making or doing whatever job it's doing. And most of the time, it needs its DNA code, okay, to be elongated. Remember, it's a double stretch. I'm just going to make this ladder. I know it's a double helix, but they open it up, and we read, or the cell reads from this DNA code to make the proteins. And so this has to be elongated. To duplicate the DNA, it has to be also elongated. So what you have is these huge, incredibly long strands of DNA, okay? And you have enzymes, guess what, and proteins that help, okay, read the DNA to make the chemical reaction. And we'll talk about, all right, translation and transcription, all the things that we do. So it has to be elongated. Now, it's duplicated in the S phase, and then it's checked for errors in this G2. And in mitosis, when we start prophase here, something happens, okay? This DNA starts coiling up on itself. And when it coils up, that's the chromosome. Okay, when it coils up, it's the chromosome. And why does it do that? Well, because if we're going to duplicate the cell, we have to make a copy. We made a copy already in the S phase, and it coils up. And guess what helps coil it up? Proteins. Hello. And the proteins are the soldiers of our genetic code. And so we have these, guess what? Enzymes. I'm sorry, chromosomes. And these chromosomes are really coiled up DNA with proteins holding together. And so prophase starts when you can, guess what? See. You can see. This is my eyeball here and eyebrows. You can actually see visibly in a cell. Here's a cell. Dark blotchy patterns of what? Chromosomes. Now you can't actually see pairs of chromosomes, but you can see the darkness due to what? Due to them coiling up on each other. In interphase, the rest of the cell cycle, okay, the cell nucleus is clear. So one of the first steps in prophase is seeing that coiled up structure. Very important. And the reason why it's coiled up, well, if I want to duplicate the cell, as I just did here, I want one of these to go to one daughter cell and another one of these chromosomes to go to another. And it's hard to do that when their DNA is elongated. Very important. Okay. Any case, that's important here. So the key here is when I say 2n equals 4, yes, there's 4, but we're going to draw actually 8 because each one of these got duplicated. So this black one here represents the duplicated one. So if I ask you to draw, and I will on the next test, 2n equals 4, then you should actually have 8 drawn, because in interphase, before we get to mitosis in the S part of the growth cycle, there was duplication of the DNA. Then it got condensed and coiled up into these tight DNA patterns, okay, DNA coils we call chromosomes. And the reason for that is we want these to separate. We could never separate spaghetti if it was nice and loose, long-stranded. But if we coil each one up tightly, we can easily move all the DNA on that chromosome. That's what a chromosome is. Okay, so very important. So when 2n equals 2, you're going to put this to, and what you're going to have here is what we call homologous chromosomes. Okay, now, the homologous chromosomes are these two, the dad and the mothers, the R and the little r. Okay, what these guys are called are sister chromatins. These are exact copies of each other. So the dad gets a copy and the mother gets an exact copy. Now in mitosis, since we're making a duplicate, okay, you're just making a duplicate pair, okay? And so you're basically, okay, going to have the same 4 that we started, all right? And so they're going to line themselves up, okay? All of these are going to line up, and if you notice, if we're going to have a sister chromatin, this is from the mother, and this is another mother, this is the dark one, and this is the blue. It should make some sense here. And this is the other one, I'll just draw 2. They're going to line up on that metaphyseal plate, right? Metaphase. That's this one, that's daddy. And that's the copy. Let's do black. And this will be a cell here. As you notice, these are going to pull away from each other, right? You're going to have the little proteins that actually pull these guys apart. The centromeres will be involved. And you're going to make an exact copy, right? One side's going to get a copy of the mother, the other get the other copy, and you're going to wind up with the same. If I'll do this with all 4, so you should be easy to see that you're going to make an exact copy. Okay, so in mitosis, because you're just making a duplicate, okay, you have sister chromatins. Now, what about meiosis? Now, meiosis only occurs in the sex cells. Has nothing to do with just regular duplication. This is how we're trying to make sexual reproduction. This is different. You're not dealing with a zygote. You're dealing with gametes. Gametes are your sex cells, and gametes have an N. So if 2n equals 4, N equals 2. So at the end of mitosis, I should have half the number. Now, what does prophase look like in mitosis? Well, it looks very similar. You're going to start, if 2n equals 4, you're going to start with 8 because you're still going to duplicate the DNA in the S phase of the growth cycle, and therefore, party people, you will have 8 as well. But this will look a little different, and this is really important. Remember, when we duplicate the cell, we want an exact copy. Why do I want an exact copy? Because if I have a cell that's making insulin in my pancreas, I want it to have the exact genetic code to keep making insulin, so it can keep making the protein that makes the chemical reactions to make the insulin keep going. I don't want to have any changes. I don't want to have no genetic diversity. Now, when I am talking about gametes and an N equals 2, we're trying to prepare sex cells that have half the what? Half the chromosomes to go into a gamete so that they can pair up with the other half from the other sex to make a full zygote. This is where I want genetic diversity, so very important. So in 2n equals 4, I'm still going to have my 4 chromosomes, mother, father, mother, father. But what's different here is that the duplicated, we'll do the duplicated one in red, is going to kind of like be connected here. And another one in red is connected here, and this one will connect here. And another one would go here, and another one would cross over here. Now what I'm showing, I'm not showing very well, is that here what we have is a tetrad. A tetrad is where we have 4 chromosomes. The red is the copy, just like mitosis, but these tetrads stick together and they have some overlapping, and that overlapping I can't say enough. I'm just going to subtly say crossing over. Yes, that crossing over is where genes of one chromosome get switched to another chromosome. If you only have 4 chromosomes, then basically you think of a regular zygote that didn't duplicate its DNA would have 4, one from the mother, one from the father, mother, father, right? And you basically could have only a certain number of combinations of offspring because all of these genes are on the same chromosome. Either I get the chromosome or not. But if I can cross over and take some of these genes and put them here and put some of these, then the combination of genes on the chromosome increase tenfold. Okay, and the number of combinations. And as you're reading in your first Nord said, you can have 80 trillion combinations of what? Of offspring due to just having different what? Variations of certain chromosomes. If you have 20, let's just make this, if we have two pairs of chromosomes for a total of 4, we can only have certain combinations of chromosomes. But with something called random fertilization, if you have one of 4, right? If you're going to make 4 gametes, so if you're going to make some gametes from 2n equals 4, okay, then you can have essentially each one of these will have what? 2, right? Of the 4, and you can have different combinations, and then they'll meet up with the mother's combination of another heritage. So because of random fertilization, some chromosomes of one father might come here, some might not come here, but because of random fertilization, not all of these gametes will make a zygote. This might have one of the genes that never get expressed because of chance. And on top of that, and the biggest point, is because of crossing over, flipping some of the genes from one chromosome to another, wow, the amount of combinations that are possible is outrageous. You would have to have 80 trillion kids in order to have a chance to have 2 kids that have the same genetic code from the same 2 parents. That means you could have identical kids with the same genetics, okay, that are not identical twins. But that would take 80 trillion times because of all of this variation. So what does this give us, party people? It gives us tremendous diversity, genetic diversity, and that's what you want in sexual reproduction. Why? Why do I want this in sexual reproduction? I do so because I want to be able to have a huge variability in my children. Why? I want people in my population, in my group of organisms living together that are the same species, to have as many different what? Alleles, okay, as possible. I want different combinations. The only way to get that is essentially from the crossing over, the independent assortment and segregation of certain genes on certain chromosomes that can be a 50-50 getting one or the other. And of course, okay, the random fertilization that can occur. So that's really important. And this last point I want to make, and this ties everything together. Why do we want increased genetic diversity? Why is it that sexual reproduction rules the landscape? Because there is an advantage to it. By having increased genetic diversity, you have a greater number of individuals in your population that by chance could survive a catastrophic event. The example I gave in class, okay, were the moths. And the moths had a certain color. They sleep on these trees, and their bark that they slept on was the same color as their wings. And birds didn't have an opportunity to eat them because they blended in. It wasn't an accident. The gray what? Moths, who have the what? Gray allele because of having the code, all right? The protein to make the chemical reaction, just by chance was advantageous. So birds, okay, this is my poor version of a bird. So birds, okay, couldn't see them and couldn't eat them. If something was other than gray, they could easily see them. So the birds ate all the organisms that weren't gray, so therefore the gray's able to live and to make more grays, and the frequency of the gray allele increased. But there has to be some in our population that's not gray. What if, and this happened, this is a true story, what if the bark became black, okay? And because of the Industrial Revolution, we had all kinds of coal and soot that sooted our forests. Now the gray what? The gray moths stood out and the birds ate them. And you would think that the entire gray moth population would die out, but because of increased genetic diversity, because of many different alleles, they were, even though they're far and few between, there were some what? Moths that had black wings, low frequency, not many, because of this genetic diversity. And so the ones that had black wings survived because they were black on black. The birds didn't eat them, and they were able to what? Reproduce and produce more. That's evolution. Evolution works because not because organisms adapt to their environment to grow what they need. They adapt based on what they already have by genetic diversity. If the organisms, if the population did not have any organisms that had black um, wings, the entire moths would have died. But because of genetic diversity given to us by sexual reproduction, because of meiosis and the random what fertilization, the crossover, and law of segregation, we have some individuals that are different, that are just sitting in the wings, maybe of low frequency, but having them there, they'll come to the forefront if they match an advantageous thing in the what? In the environment. Some of you guys answered your last question to um, the fourth question in summer assignment 2 with saying that the environment plays a role. You're darn tootin' it does. The environment will bring out this. But if this moth didn't have a low number of black winged moth, this, as most species go, would go extinct. So the reason we're here today is because of evolution, because of sexual reproduction, because of meiosis. Okay, and that's where I'm tying it in. Evolution occurred because there was a few individuals in a population of a species that existed because of this diversity that, again, in low numbers and could certainly die off, but came to the forefront and increased its numbers when that allele, that expression of the gene, okay, was more advantageous to some kind of event. And this event was the Industrial Revolution. So the Industrial Revolution made the gray moth become black, and it adapted, but it only adapted because it already had some black winged moths. They were few and far between, okay, and they were most often eaten, okay, when the bark was now gray, but they existed because of genetic diversity, and their numbers increased, and they were able to adapt because of a allele, although small in frequency, grew in numbers and was able to as the species was able to live on. So that puts hopefully everything together. All right, and this would be a good review for a test, okay, on this topic. Hope that helped.